A warm welcome to everyone. In this video presentation, we'll be discussing about formulation and evaluation of nano cocrystals based oral films of Brexpiprazole. Introduction. Psychosis is a common and functionally disruptive symptom and a defining feature of schizophrenia spectrum disorders, a common but variable feature of mood and substance use disorder. The cause of psychosis is schizophrenia, bipolar disorder, trauma, drug abuse, or its related side effects. The treatment of psychosis can be done by psychological therapy, social support, antipsychotic medications, safe care, and lifestyle. The patients who are suffering from psychotic related issues, traditional tablets and capsules are not their ideal dosage forms. It is difficult to persuade them to swallow these medications as they tend to spit them out. Drug profile. Brexpiprazole is a typical antipsychotic agent, which is a serotonin dopamine activity modulator that acts on T2, 5-HT1A, and 5-HT2A receptors. Brexpiprazole is classified under BCS class 2 drugs, that is low solubility and high permeability. Therefore, it is highly desirable to obtain new forms for improved solubility. The BCS class 2 shows low solubility over the physiological pH range, that is 1 to 7.5 pH. Thus, in vitro release is essential to determine the dissolution profile of the drug and its formulation. Formulation rationale. Co-crystals are the solids that are crystalline single-phase materials composed of two or more different molecules and are ionic compounds, generally in a stoichiometric ratios which are neither solvents nor simple salts. Pharmaceutical co-crystals alter or improve the physical properties such as solubility, hygroscopicity, compaction behavior, and dissolution. Drug and co-former on co-crystallization yields co-crystals with modulated properties. Oral disintegrating films are a type of formulations that are commonly prepared using hydrophilic polymers, enabling rapid dissolution upon contact with saliva. Advantages of ODFs include ease of transportation, ease of swallowing for geriatric and pediatrics, convenient and accurate dosing, no need of water for administration, convenient for dysplagic patients having difficulty in swallowing tablets and capsules, rapid onset of action with increased bioavailability and stability, as they avoid hepatic first-pass effects. Objectives: to overcome the solubility issues by nano-co-crystal approach and converting the same into ODFs. The study objectives include the improvement of solubility and dissolution profile of the drug, reduction in the dose-related side effects associated with oral therapy, and better patient compliance for psychotic patients. Plan of work. Objectives achieved. Selection of suitable methodology for the preparation of nano-co-crystals, screening of co-formers based on saturation solubility studies, pre-formulation studies of the drug and selected co-formers, formulation of nano-co-crystals, utilization of custom design for optimizing both the formulation and the process parameters involved, evaluation of the chosen experimental model, selection of and preparation of the optimal formulation, preliminary study of ODFs, preparation of ODFs using optimized nano-co-crystals. The evaluation of nano-crystals were performed for in vitro dissolution studies, solubility studies, as well as drug content. Evaluation of optimum formulation was done by solubility studies, in vitro dissolution studies, IR drug excipient interaction, DSC studies, as well as powder extrait diffraction. Evaluation of ODFs was done for drug polymer interaction by FTIR, in vitro dissolution studies, percentage drug content, disintegration time of the film, thickness of the prepared film, and their folding endurance. Methods and results. The pre-formulation studies of Brexpiprazole. Melting point was found to be 180 degrees Celsius and it meets the standard specified in the official limits. The initial solubility studies of Brexpiprazole showed it was soluble in methanol, dimethyl formamide, and dimethyl sulfoxide, whereas it was practically insoluble in water. The FTIR analysis showed peaks at a stretching range 3000 to 2800 for NH group, 2000 to 1650 for CH bending, 1725 to 1705 to C double bond groups, and 840 to 790 for C double bond C groups. This means that there is no interaction between the drug and excipients used here. The calibration curve was performed using both methanol and 6.8 pH buffer, and the maximum absorption was found to be 215 nanometer. Standard curve of Brexpiprazole was also done using methanol and 6.8 phosphate buffer. Screening for the selection of the co-formers. The preparation was done by anti-solvent addition method. The saturation solubility studies proved that the nicotinamide had superior solubility studies compared to other co-formers used. These are the 7 co-formers used for the saturation solubility studies. Preparation of Brexpiprazole nano cocrystals. The method of preparation used is anti-solvent addition method. Drug used is Brexpiprazole and coformer used is nicotinamide. In a solvent like methanol, coformer and drug is mixed until it reaches solubilization. In a magnetic stirrer containing water, this solution is added and kept for stirring for at least 2 to 3 hours. The obtained solution is then centrifuged at 3000 rpm for 15 minutes and is then filtered out and is kept for drying. The drying leads to the formation of co-crystals. Optimization of the formulation was done using Design Expert software V13. Factors used are coformer amount, antisolvent volume, and stirring time, whereas the responses used were solubility and dissolution rates. The upper and lower limits are set below, and the decision generated showed 7 experimental trials, as given in the table. Evaluation of optimized co-crystals. The co-crystals obtained as per the experimental trials were evaluated for solubility studies and in vitro release. Solubility showed 0.23 to 0.087 mg per ml, whereas percentage CTR at 10 minutes was 54.9 plus or minus 0.25 to 72.1 plus or minus 0.25, whereas at 120 minutes 60 plus or minus 0.26 to 78.4 plus or minus 0.48%. For the 17 formulations, the optimized solubility studies and in vitro drug release were performed, and these are the data given below. The optimum formula selection was based on prediction table and design space, and the factors influence was considered highest in terms of coformer amount and antisolvent volume, and the least in terms of stirring. Surface plot for solubility shows the lack of it and the adjusted R square and predicted R square values were better in terms of two factorial design for solubility, and for dissolution rate, adjusted and predicted R square with lack of fit was better in terms of linear model. The desirability was found to be 0.81 for the factor level where coformer was used 150 mg, antisolvent volume was used 30 ml, and stirring time was used 4 hours. The contour plot for solubility and the 3D response plot showed curvature, whereas contour plot of dissolution rate and 3D response of surface plot of dissolution showed planar or linear. The optimized formulation results. The optimized formulations, the factors and their use and their level use are mentioned below. Coformer amount 150, antisolvent volume 30, and stirring time 4. This predicted formula showed the responses where solubility mg per ml was 0.53 plus or minus 0.02 and rate of dissolution 68.5 plus or minus 0.05. SEM morphological analysis showed that the columnar crystal habits were seen with uneven surfaces. PXRD studies for the pure drug showed sharp and large number of peaks, indicating that the Brexpiprazole used is crystalline in nature, whereas the optimized formulation showed lesser intensity and the number of peaks compared to pure drug. This indicates that there is a reduction in the crystallinity when converted into co-crystal forms. Differential current calorimetry was performed for a pure drug, which showed the endothermal transition at 180 degrees Celsius, whereas optimized formulation showed endothermal peaks of 140 degrees Celsius. There is a huge improvement between these two formulations. Preparations of ODS were performed using a solvent casting method. Here, the polymer along with the suitable vehicle and drug along with the other additives like mannitol, citrate, sodium starch, glycolate, lysine, and HPMC was mixed together. These both solutions were mixed together and poured on a Petri plate and kept for overnight, drying at 50 to 60 degrees Celsius. And the dried out film was peeled out from the desiccator and was used for evaluation. The patch area of 6.2 centimeters square should contain 4 mg of Brexpiprazole was maintained during this formulation. Drug content study showed 90 plus or minus 1.65%, and it was found to be satisfactory. Film thickness of 0.027 plus or minus 0.0002 mm to 0.039 plus or minus 0.0003 mm was found to be the thickness. Folding endurance was found to be 85 plus or minus 1.25 folds. which was found to be satisfactory, and disintegrating time of 48 seconds. The in vitro dissolution study showed that respect to pure drug versus optimized cocrystals versus oral disintegrating films. The comparative in vitro dissolution studies showed major improvement. The percent CDR of ODFs 64.86 plus or minus 0.265 at 10 minutes and 71.23 plus or minus 0.125 120 minutes. The blue line in the graph indicates the pure drug. The red line indicates the oral disintegrating films, and the orange line indicates the co-crystals used. Formulation used for in vitro release study. The stability study of oral film formulation was performed in the condition both room temperature and accelerated temperature. The time period was 90 days. The observations were slight reduction in the drug content and film opacity was seen. This is the table for both physical evaluation and drug content which was performed. In conclusion, the anti-solvent addition method was found to be inexpensive to prepare co-crystals, and the combination of nanoionization and co-crystal formulation to an extent found, has shown good solubility. The custom design approach used was found to fit well, and the formulation variables and the responses selected, which were evident for the p-values. The optimum formula predicted by the model could result in nano-crystal formulas quality attributes with expected specifications, and the percent CDR in the final oral film formulation 64.86% at 10 minutes and 71.23 at 120 minutes. And this work can be extended further for in vivo biology studies too. The accelerated stability studies proved that the formulation had acceptable variation in terms of drug content and appearance. So, in conclusion, we can say that the oral disintegrating films containing nanocrystals of brexpiprazole could be suitable for as a practical approach to improve both the solubility and the overcome of the patient compliance associated issues. Thank you.